Hello lovelies, in this video the brilliant Dr. Edwards is going to go through sampling techniques for your A-level biology. Because it's not just the quadracs and transects you're maybe used to from GCSE. We need to know how to sample water, we need to know how to sample bugs that are in the air. So there are lots and lots of techniques you need to know in this video.
Hi everyone. Okay, so we're going to look at how we can measure biodiversity. So ways of measuring biodiversity to kind of calculate it, estimate it, and look at how biodiversity changes in different habitats. So in order to study the biodiversity of an ecosystem, ecologists, that's the name of the type of scientists that look at interactions in ecosystems, we need to know the abundance, and abundance just means how many organisms there are, or the distribution, and that means where they are. So how are they distributed around the habitat? And we're looking in a specific area of habitat. So we're not looking at like the ecosystem of the whole world. We're looking at kind of a specific area of habitat in a specific ecosystem. So by measuring the number of species in an area, or where the species are in an area, then we are able to investigate how various biotic and abiotic factors are affecting the population numbers. Now remember, we've talked about this before, but biotic is living and abiotic is non-living. So we're thinking about how the distribution of one organism might affect another, that would be a biotic factor. If we're thinking about how temperature affects the distribution, or where some organisms are found in a habitat, or how many are found, then that would be a non-living factor. It's impossible to count the exact number of organisms in a large area, as it would take too long, and in some places there's just too many organisms to count. For example, think about a rainforest. So if we think about in a small area of rainforest, just simply how many different organisms there would be. And if you're trying to count all of those organisms, from tiny insects all the way up to massive trees in a small area, it would just be impossible.
So instead of counting everything, we count a small amount of organisms in a small area that is known, and then we can use that to scale up and estimate how many organisms there would be in the large area. So we're going to look at different methods that we're going to use depending on whether we're sampling motile, so that means mobile, moving around, organisms that can move, and non-motile organisms. So non-motile meaning things that are stuck to rocks, like limpets, or maybe things like plants, obviously, that can't move around. We're going to use two different kind of techniques to be able to do that.
Okay, so it doesn't really matter what sampling method you're using, whether you're doing random or non-random, but it needs to be both reliable, so that you can be reliant on your results as actual truth, or as actual kind of would be able to be done again by somebody else potentially, or be able to be actual representative of the area you're looking at. So we need our kind of sampling to be robust. So what do we mean by reliable? It means the sample needs to be large. If you just sampled a really tiny number, so you just put down a couple of quadrats, or you just looked at a really tiny area, then that's not going to be very reliable, because you would have got a very small snapshot of what is the real picture, and it's not good enough to then use it as an estimation for the whole area. So the larger the area that you're sampling, so a whole beach, a whole forest, the more small areas you need to sample or need to be counted in order to make it reliable for that whole area. Representative, the sample needs to represent the area. So it can't just be taken from one small part of the whole area in one place. It needs to be representative, so the sample has to have been randomly taken from across the whole expanse of the area that you are trying to estimate. So if you've got a whole field, you can't just measure one corner. You have to measure each corner, bits in the middle. You have to have been able to sample equally across that whole large space for your sample to be considered representative of the whole, so that when we're scaling up and we're using our sample to estimate the whole population in the whole area, we make sure that our results and that estimation is going to be accurate. So the sampling method and the way you're going to sample is going to depend on what organisms you're sampling. For plants and anything that moves very small, For example, here in my picture, I've got some rocks, and on these rocks will most likely be limpets amongst this seaweed. So there will be seaweed here, and that's obviously not going to move or go anywhere. And potentially inside my area, inside my quadrat sample, there's going to be limpets. If you're not 100% sure what limpet is, it's kind of like a little shelled organism, and it lives on rocks, and it clamps down. It clamps down and sucks and attaches itself to the rock. And so they very rarely move. They will move very, very slowly, and they tend to only move around when they're grazing, which is normally when they're covered in water. So when the tide is down like this and you can see all of these exposed rocks and this area, there will probably be limpets there, and they're not going to move. So although they're an animal, they're not an animal that moves, or it's an animal that moves very slowly. You can also use a quadrat. If you're going to be looking at animals that can move around and you need to catch them, then you're going to need a different sampling method. But for the first couple of examples we're going to look at non-motile, non-moving organisms and plants. So we're going to use a quadrat. Now a quadrat looks like this. It's normally a square, sometimes wire, could be wooden frame. I've also got one here. So you can see the white square that's been laid down on the picture. It often has an internal grid, so it's got grid lines inside to mark out small areas inside, and we'll talk about why that is in a bit. You can get different sizes, from quite small ones to 1/4 metre square to all the way up to 1 metre square. So we're going to talk about how we can use this to sample in the two methods, and then we're going to talk about how we can sample animals that move. So first we're going to start with random sampling. Random sampling is a way of eliminating personal choice, which is what sometimes we also call bias, in the selection of a sample. Now, some of you may have been taught or may think that standing in a field and throwing a quadrat is random. It's not random, okay? Throwing a quadrat is not truly random because the whole area will not be equally as likely to be sampled every time you throw, because it's going to depend on how far you can throw the quadrat. So that depends on how big the quadrat is, how good you are at throwing, and also the direction you're standing in when you throw. So throwing a quadrat is not truly random for all those reasons. The more quadrats you can use to sample an area, the more reliable it will be. Remember, we've said our sample's got to be large. So if we asked how to improve the reliability of something, we can always say we can lay more quadrats. So the randomness allows us to make sure our sample's representative, remember. The number of quadrats allows it to be more reliable. So this is the method we're going to use, and it's pretty much the same for any example of when you're going to randomly sample an area to find out how many organisms are there. First of all, we're going to divide the area into a grid using measuring tapes or tape measures, same thing, placed at right angles to each other. So what we need to do, and I've got this sort of small example here, say this is my field of flowers, and I would lay my two tape measures at right angles to each other. So I've got here and here. I've laid them at right angles to each other to create a grid, and if they are a tape measure, then they will have measurements on. So they'll go up in, so say you can go up in 10 centimetres, 20 centimetres, however many. You've got coordinates, because you've literally laid like an axis down on the graph, because you've got numbers going up the y-axis effectively and along the x-axis. You can pick numbers. So you need to decide where you're going to lay your quadrats, and it has to be random, remember? So we're going to use random numbers, which means we will generate them using a random number generator, or you could pick bits of paper with the numbers on out of a hat. It doesn't matter. But each time you're going to need to pick an X and a Y, and then that will be your coordinates. So it will tell you how far along the X-axis to go, and then how far on the Y-axis to go. So say we picked 8 and 20. So I go along the X-axis 8, and then I go up my Y-axis 20, make sure I'm in the right space, and this is where I'm going to lay my quadrat. And that's how I've laid it. I've chosen an area at random by picking two random numbers and then I've used my grid to walk along 8, walk up 20, and lay my quadrat there. So once I've placed my quadrat down, I'm going to count the number of individual organisms, whichever organisms I'm looking at, or I could estimate percentage cover. So now percentage cover is a bit more subjective, because what you're doing is you are estimating. So you're estimating the percentage of your quadrat that's covered by an organism. So if, say, 50% of my quadrat, in 50% of my quadrat squares, or half of my quadrat squares, I could see a daisy, I could say that roughly percentage cover of daisies in my quadrat was 50%. But that's very subjective. It depends on how you're deciding this, and it's not as reliable. So count the numbers of each species. Now, we could just be looking at one species. In this example, I could just be wanting to count yellow flowers. So if I just want to count yellow flowers, then that's fairly easy. I'm only going to be looking at that species. I know that that's the yellow flower I'm going to be looking at. But I could be wanting to look at kind of the relationships between different organisms, or looking at the different numbers of, say, yellow flowers to purple flowers in this field. But I might need to be able to identify those plants, especially if they haven't all got flowers on. Or if I don't know, I've gone to a field and I've said, oh, I need to count the number of this species and I don't know what it looks like. So I can use a key to identify each species, which is normally a little book, a little diagram that tells you features or characteristics to look out for to identify which species is which. So I make sure when I'm counting, I'm actually counting all the species, the individuals of that species in that quadrat. Then I'm going to write my number in a table. So I count the number of species of yellow flowers or purple flowers or both in my quadrat, and then I write down those numbers in my table. And then I'm going to repeat it. So I would repeat it again, get 2 other random coordinates, go somewhere else, put the quadrat down. So say this time we went over here, I put a quadrat there, then say we went here, I put a quadrat there based on the numbers that I'm generating, and then there's one here. And I just keep repeating that until I've considered that I've got enough of a representative sample. Reliable sample, sorry. So let's see how many I can do. And let's say, we'll talk about in an example in a minute how many I've done. But once you feel like you've covered enough of the area and you've done a large enough sample, then you can stop. Then it's about calculating the mean. Remember, the mean is just another way of saying average. Number of each individuals of each species per quadrat. So for all of the quadrats, I add up the number of flowers that were yellow flowers, say, that were in each quadrat, and then divide them by the number of quadrats. Same, I could count up the number of purple flowers, the total purple flowers that were in all of my quadrats, and then divide it by the number of quadrats. Okay, let's think about just checking how we count within a quadrat. So say this is one of my quadrats that I've put down. So say it's this one over here. And I've laid it down and this is what I'm looking at. Obviously there'd be grass, there would be purple flowers in there as well. But just for a sake, let's have a quick look at how this would work. You have to decide how you're going to record the numbers of flowers in this quadrat. So what we tend to do is we won't count anything that's not completely in the quadrat. So these 2 here, I would not count because they are not totally within the square of my quadrat. So anything that's over the boundary does not count. So I'm going to have 1, 2, 3, 4, 5, 6. So the number of flowers in this quadrat is 6. And I would make sure I can keep doing that for all of my quadrats that I'm going to take. And then, as I said, so we would add up the total number. So add up the total number of flowers from each quadrat, divide it by the number of quadrats, and that gives my mean number of flowers per quadrat. If there are any extreme outliers, we tend not to ignore outliers in ecology because anything can happen. You're doing totally random distribution, so there shouldn't really be any major outliers. But if there really was something that you thought was really strange, like you accidentally dropped it on a path or something like that, and you're not looking for that kind of change in environment, then potentially you can ignore outliers when calculating the mean. But because this isn't a reaction where we're predicting a result and we know what should happen, i.e. as X increases, Y increases, this is random, it's nature. It's going to be different. Okay, so non-random sampling. So a transect is a way that scientists can look at the distribution, remember that's where organisms are, of a species and how it changes as the habitat changes. It's useful for investigating how biotic or abiotic factors affect population numbers. Now, random sampling, you can do that. You could sample a field that has been mown and a field that has not been mown. Or you could sample a bit of desert that has really high temperatures at some point, and then you could sample a bit of desert or similar dry environment with low temperatures. And then you can just compare the numbers. What we're talking about here is about change within one habitat. So we still use a quadrat, but we're placing it at regular intervals along a single tape measure this time, so we're not using a grid. And that single tape measure goes through a changing part of the habitat. And because we're placing the quadrat at regular intervals along the tape measure, we're not randomly placing it, therefore it's systematic sampling. So this is the method we're going to use. So we're going to take a tape measure and we're going to lay it across the area. Now it should be going across an area of change. So I've got a couple of examples. We could be doing it from right underneath a shady tree to far away from the shady tree. So in this example, we'd be looking at changing light levels. And that would be like our abiotic factor that we think is going to affect it. We could also be looking at changing or how something changes as it gets closer to or further away from a river. So in this case, we'd be looking at water concentration, or moisture levels. So that soil is going to get wetter and wetter as we get closer to the water. And so we could be looking at how the plant's distribution changes as we get closer to it. So we know the habitat's changing along our transect, and we're trying to look at how that's affecting how many of each species are going to be there. So we need to always start with placing a quadrat at 0 meters. So at the very, very start. So it depends on what direction you're going in, whether you're going towards or away from. But for example, 0 meters could be here by the bottom of the tree, it could be here by the start of the lake. Place your quadrat down, do exactly the same. So given what we've just talked about, 3, 4, 5, exactly the same as what we've just previously looked at, how we count species in the quadrat. And then you're going to move the quadrat along the tape at a set distance. So you will decide how far along you're going to put your tape. Are you going to put it every 2 meters, 10 meters, 20 meters, depends on how long the transect is. You will decide where to put it. Then 5 meters, then 10 meters. It needs to be the same. Say for the river, we did it slightly differently. So we did 0 meters, then we did 2 meters, then we did 4 meters. As long as it's regular intervals, so every 2, every 5, every 10 meters, it doesn't matter. But the intervals need to be regular. So remember, this is not random. You then repeat exactly what you've done every single time you do a quadrat, all along the transect. And then this is the thing that is very important that people often forget. In this case, we can't just add more quadrats. What we need to do is we need to repeat the transect. So it's got to be a slightly different position so the quadrats aren't overlapping, but you need to repeat it so you're getting the same thing. So again, at 0 meters from the lake, again at 2 meters from the lake, river, again at 4 meters from the river. So each time what I've done is I've placed another transect. So I'm getting similar data, but the quadrats aren't overlapping each other. So I'm not getting the same data, but it's still in the same area, it's still in the same kind of direction, and the distances will be the same. And that's how we're going to get our average. So we will average together the 0 meter quadrats. We will average together the 2 meter quadrats and the 4 meter quadrats. Same for here, we'd average the zeros, we'd average all the 5s, and we'd average all the 10 meters. And then that would give us our more reliable sample by doing it more than once. Okay, so that's how you would make it more reliable. You'd repeat the transect in a similar direction or in a similar area.
Okay, so this is obviously an example of an interrupted transect. The pros of an interrupted transect are that it's faster. The cons of an interrupted transect are that it's not as informative as if you did a belt transect where you're going each quadrant next to each other. The other thing is that you're going to need other equipment to be able to do this. Really, if you're looking at a changing condition, you should be measuring that changing condition. So you need some form of equipment for measuring the sort of the abiotic factor or the habitat change that you're going to look at. So things like a pH meter if you're thinking about change of pH, or maybe it's about salinity. So you can have a reader that will measure the amount of salt content, or you could simply do stuff like take samples from each quadrat of soil or water, for example, and take them back to the lab and do that analysis later. But ideally, you'd use something like a pH meter when you're there in the soil, a light meter or a lux meter to measure light intensity right down where your plants are, a moisture meter to measure how much moisture content there is in the soil, and all of that would then help you get an idea of kind of the abiotic factors that are happening in each quadrat. So you'd have to take the readings from the same point, so say the centre point of each quadrat for each of your measurements, and then you could plot that data alongside your abundance data, and that will give you kind of that pattern or maybe a correlation if that's what you're looking for. And then you can obviously show how that feature might actually affect the abundance of whatever organisms you're looking at. Okay, so when we're using a quadrat, we can obviously count every individual that's in that quadrat. Sometimes that's harder to do than others. There's still too many. You can't really see or separate individuals, especially if you're looking at something like grass. You can't separate, the blades of grass aren't always going to be from separate organisms, for example. Or if you're trying to do it quickly and it would take too long to count all of them, there's sort of different ways that you can use a quadrat to kind of estimate population size. So the first one is percentage frequency. So the probability that a species will be found in a single quadrat. So we work that out by doing the number of quadrats that a species was found in divided by the total number of quadrats times 100. So for example, 30 quadrats are randomly placed in two different fields. Maybe one's shadier than the other. Maybe one is the grass is cut in one and it's not in the other. Maybe they've got two different sort of crops growing in the fields. And in one field, daisies were found in 18 out of the 30 quadrats. So it's just whether they're present or absent. That's all it is. Did you have at least one individual in that quadrat? And in the other field, daisies were only found in 6 out of the 30 quadrats. So then we can obviously do the number of quadrats the species was found in, divided by the total number, which was 30 for each one, and then that gives us a percentage. So 18 out of 30 times 100 gives us 60%. The percentage cover in field 2, 6 out of 30 times 100 gives us 20%. So the other way we can estimate sort of how much space a species is taking up in a certain area is using percentage cover. So percentage cover is faster, but it's more subjective because you are not actually really counting or measuring. You're using a visual estimate of an area of a quadrat that's covered by a species. So it tends to be overestimated for flowering plants, where you can think about they've got sort of broader flowers and leaves covering a certain area. And then also underestimated for plants that grow very close to the ground, ones that you can't see so easily. So it's often easy to do this if you use quite a large quadrat, a quadrat that will be 10 by 10 squares. Because then you've got 100 squares, and then that means that you can just see how many squares are covered in total in area by the species, how many sort of rough squares you can find that species in, and then out of 100 that will give us a nice easy percentage. So here's an example. I've got 3 different species of plant. They're all got different coloured flowers, and then I also have grass as well in my quadrat. And so that's obviously the grass is just the green, so how much is just containing the grass, and then you've got red, pink and white flowers of the different species. So I've roughly counted sort of, if it's a whole square, it's obviously covered, it definitely counts. And then I've had to use my kind of subjective judgment as to whether something's covering the majority of square or not, and sort of a rough estimate of how it is. And obviously this makes up roughly just under 100%, so it's not perfect. And obviously, technically there's grass in every square. It's just about where the grass is the majority and where it's the minority. So percentage coverage, you can see, is a way of doing it quite quickly and getting kind of a general idea of how much area a species is covering. But it's not going to be as accurate as actually counting them individually or even doing percentage frequency.
Okay, so now let's think about if we're trying to look at the distribution of a species or the abundance, how much of a species there is in a habitat, but it moves around. We're going to have to be able to catch them. So we have to use specific equipment in order to catch them so that we can then start counting them. There's lots of methods and it's going to depend on what you're catching and where you're catching it. Does it live in the water? Does it live in the trees? Is it on the ground? Is it going to live in grass? And how big is the organism? What type of organism? Is it going to fly? Is it crawling? Is it big? Will it be attracted by food? All sorts. But you need to think about the fact that you've got to do it ethically, so we're not trying to harm them or stress the animals too much. And so we've got some examples. First one is a pooter, a funny name, I know. It's used to collect some small insects by sucking them into a little chamber. So it looks a bit odd, but the way it kind of works is you have a little kind of canister, often with a lid, and it will have 2 tubes coming off of it. And there will be a little filter here on one of these tubes. So what you do is you suck using your mouth, you literally suck like a straw on one end, and that creates a little vacuum in here. And so then you put this close to a little insect, a little bug, and what will happen is they will get sucked into this little chamber, and then they won't be able to climb up and crawl out. So in an area, you've got a quadrat, say, or you're looking at a little area and you need to collect some, you will be sampling an area, so you'll be kind of sucking all the little insects you see, and then you can count them in here, and then you can take the lid off once you're done and free them. A pitfall trap is a small trap where you can dig a small hole, and it's used for kind of very small organisms, could be kind of crawly insects that would be maybe too big to be sucked up into a pooter, for example, or you want to be able to leave the trap over time and come back to it. And that's going to be useful when we're going to talk about how we calculate this. So it can just be simple as you dug a little hole, you have covered it with leaves or something that will easily fall into the hole, so that a bug will walk along and the weight of it will cause it to fall into there. But this is deep enough that they can't climb back out again. And that's all a pitfall trap is. Sweep nets, so these are just really large nets, often with very, very fine mesh. So something that so they normally have really long kind of area on them, and then a big kind of area that you can then drag across, say, long grass. Often they can have tall handles so that you can use them in the air to catch flying insects. Or you might need a really big net if you're going to go and do some sampling in water, which you can do, as long as you hold the net in for the same amount of time each time, sweep it around the same amount of time and use a net with the same size, then you can sample in water as well.
This is really important when we're thinking about making sure that what we're doing is sort of fair and accurate. You need to make sure that if you're sampling water, for example, from different places in a river, you need to hold the net that was the same size, so you're sampling the same area / volume, and you need to hold it in the same part of the stream or for the same depth of the stream for the same amount of time. So an idea is that the flow of the water means that you've measured the same volume of water has gone into your net before you scooped it up, so that then you can compare your samples that you've taken at different points of the river, because you sampled sort of the same volume of water each time by holding the same size net in a similar depth and for the same amount of time, and then that allowed you to collect similar sort of volume of water, which then you can compare the number of species in each of your samples of your nets, because you did sort of sample the same area, for want of a better word, which is harder to do in water. For the others, obviously, you'd be sampling, you'd when you saw insects, you'd be sampling them from the same area. Again, it counts on you seeing them with the pooter, so you have to visualize the insects in order to suck it up. Or you can obviously just run the edge of the pooter along a certain area and continuously suck, and then see what insects you collect after that amount of time. So say you sample for a set amount of time in the same surface area of somewhere in order to get those insects. And then for the pitfall traps, again, same size, same area of the trap, try and disguise it in the same way, using the same materials. If you were going to use food to attract organisms, you'd have to use the same amount of food, the same food. Trying to think about all these ways that you can make these collection methods very repeatable and able to show that when you've sampled various different areas, if you're doing areas and comparing them with the same methods, that you've done the same things each time to make sure that your results can be compared. Okay, so the other thing we have to think about, and we sort of talked about this at the start, was to think about the ethical considerations. So some people think that sampling or collecting organisms, full stop, might cause stress to the organism, so it's unethical to do it in the first place. So in order to try and make sure we're doing a really good job of collecting organisms, if this is something you're going to do for one of your practical tasks, similar to how we said with the woodlice when we did the behaviour experiment, you should keep handling to a minimum. You're really only collecting them to count them and to maybe identify what species you've got. So you don't need to keep them for any length of time. You don't really need to hold them, play with them, touch them or anything. You can just kind of look, count, take photos to help with identification, and then release. So all organisms should be treated carefully and with respect. If animals are put under too much stress during capture, it could reduce their chances of survival after you release them back. So that's obviously something we don't want to do. You don't want to hinder this organism's life by stressing it out and making it more easy to be predated upon, for example. It could cause them to be wary. So wary and avoid traps in the future. So if you're doing the mark recapture technique, which we're going to look at in a second, you obviously want them to potentially come back to the trap in the future, so that can affect the accuracy of your estimations of population size if they are now too scared to come near something like a pitfall trap, for example. Okay, so how do we actually use our equipment to catch our organisms, and then once we've caught them, how do we actually use that to estimate population size? Well, we're going to use something called the mark, release, recapture technique. So we take a sample from the population using appropriate equipment. So whether it's a net, whether it's a pitfall trap, however you've sampled a population of moving organisms, and you're going to count obviously the number of species of each species in that sample. Then you mark them in some way. It's got to be harmless. It's got to be something non-toxic. It's got to be something that isn't going to hurt them. You're not kind of punching a hole in their ear or anything like that. It's got to be something that can't damage them in any way and that doesn't hinder them in any way. So for a spot of paint or nail varnish, for example, on a hard substance like a shell or something like that, or identification tags, so a loose tag around the leg, especially used for things like birds. And once you've done that, you're going to release them back into the habitat. So you will have recorded how many marked animals there are in the total population, because you've taken a sample, you've marked them, and then you've put them back into the whole total population, and you know how many there are, or there should be. Then you need to wait long enough for those marked animals to have redispersed and mixed back in randomly with the rest of their population within that habitat, within that area. So it's at least 24 hours. It could be longer. Sometimes it could be weeks, you could be months and coming back. Obviously, you have to think about seasons changing and things like that, so it can't be too long, but you need to have given them time enough for them to have redistributed and mixed up randomly. Then you'll do exactly the same thing. So you'll use exactly the same equipment, the exact same technique to take another sample from ideally the same area, same place that you laid the previous trap or that you used the net or whatever it was. And in that sample, you will need to record two things, not just the number of animals that you get, but also the number of marked animals that you have got within that total. So the number of recaptured animals, because they will be ones that you caught last time, marked and released. And then this is how we're going to work out an estimate of population size, because the proportion of marked to unmarked animals in the population should be similar or the same as the proportion of marked to unmarked animals in the second sample you collected. So then you can calculate it using the following formula. So by taking the number that you caught in the first sample, multiplied by the number that you caught in the second sample, so what was your kind of sample size from the two samples, divided by the number that were marked in the second sample. So that roughly gives you your proportion, and then that has now, we've basically given a population size here. So this is the way we'd use that equipment in order to catch organisms in a way that then we can use it to estimate or calculate population size of organisms that are moving around in a habitat. So there are some problems with using this technique to estimate population size, because it relies on some assumptions, which are obviously some are realistic assumptions, or assumptions that we can be confident that can happen, and some that can't. So it relies on the idea that your population is closed. So in your habitat that you've sampled, your population, the population itself, the total number of organisms of the population stays the same. That would mean no migration out or into the population, so no organisms leave, no organisms come in, and that there's no births or deaths that occur. And this just has to be in the time between the first time you take your sample and the second time you take your sample. This is something that obviously can't be controlled. The sooner your sample is taken from between first and second, the less time there is, then you can kind of be a bit more confident that this is less likely to have happened, but you can't control it at all. And also there's things like actual death, so the chance of organisms actually dying. Then there's also things to think about, like, oh, what if that organism got trapped somewhere? Or what if that organism has flown away? Or what if that organism can't be captured for other reasons because it's a butterfly that has, or a caterpillar that's now gone into a cocoon, so it's not moving anymore, so therefore it can't be captured. There's sort of other things that can limit how the chances of you getting the same number of organisms to sample each time. It also assumes, similar to what we've just been saying, that all members of the population mix randomly. So when you put them back, they've completely randomly mixed up. They haven't stayed in the area near to where the trap is. They've sort of gone back into the population and mixed. And so that each member of the population has an equal chance of being captured every time. So when you go and do your sample, obviously you've collected a certain number of organisms, you put them back, you need them to go and completely mix randomly, so that when you go and do your second sample, you're not just resampling the same ones that you've already done. Again, it relies on the fact that all the animals will behave exactly the same, that they will disperse, that you've left enough time for them to disperse, and it also relies on certain animals behaving in a certain way. So, for example, males and females are often going to behave differently, especially if it's around springtime and the females are caring for young. So they're not going to go exploring, they're not going to leave nests, they're not going to leave their children, or they might not be able to move as far because if they've got offspring. They're not going to be able to take risks. They won't be demonstrating risky behaviour, so they're not going to sort of go into somewhere they're unfamiliar with. So they're less likely to be caught. So in reality, if you're only really sampling males at a certain time, you're not sampling all of the population, or you're not able to equally chance of sampling all of the population. Again, this is something you can't necessarily control. That you assume that the marks that you've put on, or the tags that you've put on, have not been rubbed off or lost or removed between captures, so that you definitely know that if you marked them before, they will still be marked when you go back. This is something easier and something maybe that you can try and control a bit more. But you shouldn't try, in order to do this, use anything that is really sort of sticky or really toxic, or anything that could be sort of super waterproof and therefore is a danger to the animals. And really think about what you're using in order to tag them ethically, as well as trying to make sure it doesn't rub off. Also, you're assuming that your mark or tag will not harm the animal or affect its survival in any other way. It shouldn't slow it down, its movement. It shouldn't affect its movement. It shouldn't likely change its behaviour. It shouldn't make it be, shunned by the other organisms and like left out and then not shared food or whatever, or not given access to certain places. It shouldn't make them more likely to be predated. So, for example, if you put bright paint on a prey species that is normally going to be camouflage, you've sort of ruined that in some way. And so then predators are more likely to be able to see it, and therefore they're more likely to catch it and eat it. which means you've increased the chances of your marked organisms being killed, predated upon, before you do your second sampling, and so therefore then you've got an unfair test there, because your marked ones were more likely to die, so there's going to be fewer of them in the population when you sample the second time. So it's one of those things, again, you can try and do the best you can to control this, but you're not always going to be able to account for animals' behaviour. Okay, so that is sampling, all of it, so from moving organisms to non-moving organisms, how to use a quadrat, how to estimate percentage cover or percentage frequency of a quadrat, how to do transects, how to randomly sample using the quadrat. And thinking about all of those practical things we've considered. So timing, size of the equipment you're using, where you're using it, when you're using it, when are you sampling, what time of year, what time of day. Thinking about the mark recapture and how to label or tag your organisms, and what considerations have you got to think about. Understanding that this method is not always going to be super reliable because of these assumptions that we just can't control. All of these, and this is most likely going to be given to you in a data question or a practical question where they've done some form of experiment. There'll be some data. They'll maybe ask, how could you use the mark recapture technique to measure the abundance of this organism? They could give you data that's been already used. They could ask you to talk about what the limitations of this method are. They could ask you about how to work out percentage frequency from a question. There's loads of things they can ask you, and it's mostly just about thinking, having your practical head on, and thinking about all of the different variables that you can kind of discuss here, as well as the methods as well, and sort of the pros and cons of each, and being able to evaluate the methods as well.
Ouch! This is why in some videos I'll explain scratches.